Jon Snow, and Jonas Salk. That is the place that Dr. Corbett has earned in history. But if past record is an indication of the future, she may yet be denied that place in history. Time will tell. I offered the remarkable story of Dr. Corbett as an example of an African-American woman making a great scientific contribution of global impact. Her story is a story for celebration as part of the history of Black people who have made monumental contributions to science and medicine over the last 2 centuries. That history of the contribution and accomplishment of great Black scientists is not proclaimed loudly enough, and it is not proclaimed often enough. And because the history of great Black scientists is not loudly and often proclaimed, the majority of humankind is either genuinely or willfully ignorant of the history. In the midst of these turbulent times, let us be inspired by the contributions of Black scholars and innovators, people like Dr. Kizzy Corbett, and people like our keynote speaker today, Dr. Consuelo Wilkins. Dr. Kizzy Corbett, Dr. Consuelo Wilkins, and many other famed Black scientists should be our narrative. And it is our duty, it is our obligation, I believe, to control that narrative. Controlling our narrative is what we are doing here today. Thank you.
Thank you, Dr. Ojo, for sharing those stories with us. You as well are an excellent scientist and a great mentor to many young scientists of color. And we really appreciate you joining us today and for your heartwarming remarks.
Thank you.
Well, good morning, everyone, and welcome again to Research Day of our KU Medical Center 2021 Black History Month celebration. Before we get started, we wish to announce that we have our wonderful sign language interpreters, Christina Godinez and Chris Castor, with us today. If you need to view a sign language interpreter, please switch your screen now to gallery view by clicking the icon at the top right-hand corner of the screen. If you have trouble finding the icon, please let us know by the Zoom chat. We also need everyone to please mute your microphones unless you are currently presenting. I am Diane Mahoney from the School of Nursing. I will moderate the first segment of this morning's session. I am serving as co-chair of Research Day Organizing Committee this year. Our subcommittee also includes our committee co-chair, Dr. Hal Zhu from the School of Health Professions, Antoinette Redmond, Diversity, Equity, and Inclusion, Dr. Babalola Fasharu, Population Health, Jessica Idowu, Pharmacology, Toxicology, and Therapeutics, Suma Suswaram, the Interim Campus Program for Communication Sciences and Disorders, Dr. Michelle Redmond, Population Health, Dr. Eric Badoni, Alzheimer's Disease Center, Mary Beth Warren, Executive Director of Continuing Education and Professional Development, and Captain Donald Holland, Strategic Services. Working together with the Black History Month Planning Committee, our team has assembled today's program to showcase the various research activities being performed here at KU Medical Center by our faculty, our staff, and students of African ancestry. The theme of our KU Medical Center's Black History Month celebration is truth, and that represents transcendent, resilient, uplifting, transforming, and history makers. The organizers would like to do a polling for today's Research Day activities. and we will provide all participants the link to the REDCap survey via chat between the keynote presentation and the closing remarks. Please feel free to fill it out so we can improve our program next year. Here's a reminder for our speakers. Please pay attention to your chat box before and after your presentation. For our audience, please submit your questions by using the chat box and address your question to that particular speaker. Unfortunately today, we will not have time for a Q&A session. Of course, you can always contact the speaker directly via email, and they will also provide their email information to you. We will begin with 12 short presentations related to a variety of research topics. With that, I welcome Dr. Joan Lewis-Wamby to start our first short talk session, highlighting basic science research. The title of this presentation is Identifying and Targeting a Novel Molecular Marker in Triple-Negative Breast Cancer.
Thank you all. I'll begin by providing a brief
Joan, you're actually on mute.
She's supposed to be. It's recorded.
Okay, thank you.
Oh. Be diagnosed with 44,000 deaths. There are 3 subtypes of breast cancer: ER positive, HER2 positive, and triple negative. ER positive breast cancer represents the majority of breast cancer, 60 to 80%, and those cancers are usually treated with targeted therapy, endocrine therapy, and patients have good prognosis. HER2 positive breast cancer is also treated with targeted therapy that targets the HER2 receptor, and again, those patients have good prognosis. Patients who are diagnosed with triple negative breast cancer, those tumors lack expression of the 3 receptors that I just mentioned. So they're ER, PR, and HER2 negative. And so as a result of lacking these receptors, there are no targeted therapies available for triple negative. So patients who are diagnosed with triple negative disease, they're usually treated with systemic chemotherapy. And so the overall response rate for those patients is very poor. And so most patients will eventually progress to metastatic disease. And what we also notice is that triple negative breast cancer is disproportionately higher in young African American women compared to Caucasian women. And these women have a higher mortality rate compared to other ethnic groups. So our lab is really interested in understanding what is driving the triple negative breast cancer disease, and if we can identify a specific target that we can develop for therapy. So what we did for our study is we were able to collect triple negative breast cancer tumor sample from the biosample repository here at KUMC. We collected a total of 100 samples. We had 50 of Caucasian women and 50 from African American women, as well as normal breast tissue. And we looked at this protein of interest, ifidem1, which is a transmembrane protein that's highly induced by interferon, and it's known to block viral replication in normal cells. And it's also important for stimulating the immune system. And so we're interested in this protein because it's a protein that we've been studying for a while. And what we noticed was that this protein seems to be uniquely overexpressed in triple-negative breast cancer tumor samples from African American patients compared to Caucasian patients, and it's not expressed in normal breast tissue. And its overexpression is associated with worse overall prognosis and survival for patient, as demonstrated in panel B. We were also able to collect triple negative breast cancer cell lines from African American patients as well as Caucasian patients. And we noticed that IFIDM1 is also overexpressed in triple negative breast cancer cells derived from African American patients, but not expressed in triple negative cells derived from Caucasian patients, which is consistent with our clinical data. So to get a better understanding of the function of IFITM1 in our cells, we developed this CRISPR system in our labs, which provides us the opportunity to genetically knock out the IFIDM1 gene in our cells of interest. And then we took those cells, which are modified, and so we have cells that express IFIDM1, which is the Cas9 control cells, and cells that do not express IFIDM1, and that's the IFIDM1 knockout cells. We were able to put those cells, inject them into the mammary fat pad of mice that are genetically engineered for these types of in vivo studies, and to look at whether or not the expression of IFIDM1, if it affects how well these animals are able to develop tumors. And what we notice in panel A, B, and C is that when you inject these mice with these modified cells, that knocking out IFIDM1 significantly inhibits the ability of these animals to form these triple negative tumors in vivo. So it's clear from this data that the overexpression of IFIDM1 that we see in these triple negative cells derived from African American women, that these cells, when you put them into animals, can form tumors, and that if you knock out IFIDM1, you're able to significantly reduce the tumor size in these animals, which suggests that IFIDM1 overexpression promotes tumor growth for triple negative, and that when it's not present, that the tumors are not able to grow. So overall, I think our finding is clinically important. It shows that our target IFIDM1 is overexpressed in triple negative breast cancer tumor samples from African American patients as well as cell lines, and that if you inhibit IFIDM1, that you can actually significantly reduce the growth of triple negative tumors in animals. So overall, we think that IFIDM1 can be potentially an important therapeutic target for the disease. So I'd just like to acknowledge the people in the lab. Everyone who's contributed to this project are shown in the picture. My funding source, my collaborators, and of course the core facilities here at KU. Thank you.
Thank you so much, Dr. Lewis Mwambi. We will now move on to our next talk by Jeff T. Akapo. The title of this presentation is Mass Spectrometry Imaging of Acetaminophen and its Metabolites in the Kidney.
Traditionally, methods for probing the spatial distribution of drugs have generally relied on the use of radiolabels or antibodies. However, these are not specific enough to secondary metabolites, which can accumulate in tissues and be very toxic. MSR, or mass spectrometry imaging, is the only technique, a very powerful technique, that can distinguish between drugs, the secondary metabolites, and endogenous compounds. Not only can MSI measure the abundance of compound, but it can also map the spatial distribution of these compounds using a very powerful technique called DAISY. This MS imaging technique utilizes a capillary that has a jet spray combined with liquid nitrogen that allows to desorb analytes of interest that are then sent to the mass spectrometer, which acquires the data by measuring the intensity of individual analytes based on their mass to charge ratio. This data is then used to reconstruct the ion image of the intensity of these individual metabolites, and this reconstruction can be then combined to form the final ion image. Our lab focuses on studying the cellular and molecular mechanisms of acetaminophen overdose. Thanks to many mechanistic studies, we understand the pathophysiological events of acetaminophen overdose. And so when acetaminophen is entering the body, the majority of acetaminophen is glucurinated and excreted in the kidney. So The aim of this study was to map acetaminophen or APAP and APAP glucuronide on kidney tissue section. To do so, mice were treated with 900 milligram per kilogram APAP, and kidney tissue sections were collected and cryosectioned at 12 micron thickness. These tissue sections were collected 30 minutes after acetaminophen, because this is the time point that we know that the majority of acetaminophen are at the peak level. DAISY analysis of APAP and APAP glucuronides were also conducted, and the advantage here is that DAISY mass spectrometry imaging is a label-free technique that requires no pre-staining. And these are the results. We were able to spatially map APAP and APAP glucuronide at 50 micron pixel size on kidney tissue sections. At the lower bottom right here, you can see a representative H&E image of the kidney tissue section, which shows the outer medulla and the inner medulla. At the top, you can see APAP, the parent compound, with its protonated adduct, its associated adduct, and its potassiated adduct, as well as its metabolites, APAP glucuronide, and the lipid that is being used as an internal standard. When you combine and overlay these images, what you can see is a concentration gradient that goes from low APAP and APAP glucuronide in the cortex to concentrated APAP and APAP glucuronide in the inner medulla. And what we expect is that we see the majority of acetaminophen metabolism happening in the cortex, which we've shown previously. So what is the impact of our study? We are currently conducting a clinical trial with 4-methylpyrazole or 4-MP, which is an antidote against acetaminophen toxicity. And it protects by inhibiting CYP2A1, which is the major drug metabolizing enzyme of APAP present in the kidney. And so we want to use MSI or mass spectrometry imaging, which will help with studying the efficacy of 4-methylpyrazole against APAP toxicity. This is at the cutting edge of what a mass spectrometer can do. We are pushing the frontier to be able to use mass spectrometry imaging to study the toxic effects of acetaminophen and the potential benefit effect of 4-methylpyrazole. With that, I would like to acknowledge my mentor, our collaborators, my lab members, and the fundings. Thank you.
Thank you, Jeff T.
Thank you for
Now we will move on to our next talk by Jessica Idowu. The title of this presentation is Hepatocellular Uptake, Transporters, and the Functional Consequences of Altered Membrane Composition.
Thank you for the introduction. In the last few years, one topic of interest has been obesity in both kids and adults, which is understandable considering the CDC approximates that 40% of adults and 20% of children are obese. The cause of what has been termed the obesity epidemic includes inactivity, diet, and eating habits, but the risk factors are numerous. One prevalent risk factor is non-alcoholic fatty liver disease, or NAFLD. NAFLD is an umbrella term for liver malfunctions unrelated to alcohol consumption. Two characteristic traits of NAFLD are an accumulation of fat in the liver and increased free cholesterol levels. Free cholesterol is found primarily in the plasma membrane of cells, and cholesterol's presence has the ability to alter plasma membrane fluidity when its levels are changed. As a consequence, the function of transmembrane proteins can be affected. In my lab, we are interested in transporter proteins expressed in the membrane, which are represented in the hepatocyte figure. In particular, though, I am interested in investigating how the function and expression of the transporters NTCP, the sodium taurocholate co-transporting polypeptide, and OCT1, the organic cation transporter 1, are affected by changes in free cholesterol levels in the plasma membrane. NTCP and OCT1 are responsible for the uptake of bile acids and drugs like statins, metformin, and acyclovir. My research is important because changes in free cholesterol can affect transporter function, which means that drug dosing may need to be adjusted for changes in transporter activity from alterations in cholesterol levels. For example, if the transporter function is decreased, drugs may stay in circulation longer, potentially causing adverse reactions. In the same manner, if the transporter function is increased, drugs may be cleared from the body faster, resulting in diminished drug effects. In order to perform the aforementioned research, HEC293 cells that stably express the transporter of interest are used. With those cells, the transporter function is determined using an uptake assay. The uptake assay involves incubating the cells with a radiolabeled model substrate for the transporter. The transporter will uptake the substrate until the process is halted, after which point a beta counter will determine how much of the substrate was taken into the cells, and the value will be normalized for protein. In order to determine the effects of changing the free cholesterol levels in the membrane, two treatments will be used. One is cholesterol that is complex with methyl beta cyclodextrin in order to allow for the cholesterol to readily enter the cell membrane. The other treatment is methyl beta cyclodextrin, which is used to remove cholesterol since the molecule has a hydrophobic core that readily binds nonpolar substances. The results of the treatments to either remove or add cholesterol to the plasma membrane can be witnessed in this slide. On the left, you have NTCP uptake of the model substrate taurocholate. The graphs all have uptake as percent of control with the treatments on the x-axis. When adding cholesterol, you can see that there is a decrease in NTCP function. However, if you remove cholesterol, NTCP function increases. Now, if you look over to the right, you can see OCT1 uptake of the model substrate MPP+. In these graphs with the same x and y axes, you can see that adding cholesterol once again causes a decrease in OCT1 function. This time, though, removing cholesterol also causes a decrease in OCT1 function. Altogether, these results allow for the conclusion that changing the free cholesterol levels in the plasma membrane has different effects based upon the transporter being looked at. This, as I mentioned previously, can cause drugs to stay in the system longer due to a decrease in function or exit the system faster with an increase in function. I would like to take a second to acknowledge the help of my mentor and lab members, the Pharm Tox department, and my committee members. Thank you for your time.
Thank you, Jessica. We will now move on to our next talk by Wendina Parks. The title of this presentation is Implications of ROS-Mediated Hyaluronan Loss in Reproductive Aging of the Mammalian Ovary.
The ovary is a female gonad, and the main functions of the ovary includes gametogenesis to produce a fertilizable egg, as well as hormone secretions such as estrogen and progesterone. The ovary is the first organ to begin aging in the body, and ovarian aging is associated with a decreased number of follicles, as well as increased fraction of poor quality follicles or oocytes. Menopause, the point at which fertility ceases, usually occurs at 50 years of age. Studying reproductive aging is clinically relevant as globally more women are delaying childbearing. This is due in part to women living longer thanks to advances in the development of antibiotics and vaccines leading to increased lifespan since the 1900s, and because more women seek education and careers outside of the home. Menopause at 50 indicates that women live around half their lives with suboptimal hormones and reduced follicle quantity and quality. However, we can appreciate that the follicles do not develop in isolation. Instead, they grow and develop with an underappreciated ovarian compartment, referred to as the ovarian stroma. Therefore, while other researchers focus on the follicles with respect to reproductive aging, our lab strives to understand age-associated changes in the stroma. Specifically, our lab is interested in the role of the extracellular matrix, a major component of the stroma in reproductive aging. In particular, we are interested in understanding the role that hyaluronan and ECM glycosaminoglycan plays in ovarian reproductive aging. This will be investigated in two aims. In aim 1, we will be investigating the extent to which ovarian stromal hyaluronan content contributes to reproductive aging phenotypes. Using the hyaluronan binding protein assay, which is an assay that detects hyaluronan by binding to the G1 link domain of aggrecan, we detected hyaluronan in reproductively young and old mouse ovaries. We observed that with reproductive age, there is a decrease of hyaluronan in the stroma, including a specific cell compartment called the theca cell layer, which is associated with increased collagen accumulation and tissue stiffness, suggesting that hyaluronan might play an important role in maintaining normal ovarian function, and its loss may negatively impact reproductive aging phenotypes. Hyaluronan is degraded by two mechanisms, either enzymatically through the action of hyaluronidases or through a non-enzymatic process mediated by reactive oxygen species, which I will abbreviate ROS. When we investigated the hyaluronidases, only HAL1 significantly increased with age, but it was not the most abundantly expressed hyaluronidase. Therefore, since ROS is produced during normal cell metabolism, it is ubiquitously produced in tissues, and because ROS increases with reproductive age, we hypothesize that ROS is the main contributor to aging-associated ovarian hyaluronan loss. Therefore, aim 2 is to determine the extent to which ROS-mediated hyaluronan depolymerization is a mechanism by which stromal hyaluronan is lost. To generate some preliminary data for our second aim, we use reproductively young and old mouse and stroma tissue, which is ovarian tissue after removing follicles, and we use this to measure mRNA levels of oxidative stress and antioxidant response genes by qPCR. 4-hydroxynonenal, which I will refer to as 4-HNE, 4-HNE staining was used to measure lipid peroxidation, a surrogate marker of ROS production. From our qPCR result, we observed an increase in the transcript encoding oxidant-producing enzymes, while transcript encoding antioxidant-producing enzymes were differentially regulated. By analyzing 4-HNE in the different ovarian subcompartments, we observed increased lipid peroxidation in the theca cell layer, which is a stromal cell compartment. As I mentioned earlier, hyaluronan decreases with age in the ovarian stroma, and there is significant hyaluronan loss in the theca cell layer, a component of the ovarian stroma. The increased theca cell lipid peroxidation and loss of hyaluronan in the theca cell layer suggests that there might be a positive correlation between ROS and hyaluronan. These findings are important because while substantial research has been completed on reproductive aging by focusing on the follicle, very few treatment options have been developed. Therefore, by shifting the focus to the ovarian stroma and understanding mechanisms that promote ROS-mediated hyaluronan depolymerization, such as the use of antioxidants, we hope to find treatment options and improve fertility. Thank you for this opportunity to tell you a little bit about my dissertation research. I could not be more honored to speak in this forum celebrating Black History Month. I would be happy to answer any questions you have at the end of the session or later via email.
Thank you very much, Wendina. Our next speaker is Dr. Kendrick Waite. The title of this presentation is Molecular Regulation of Proteasome Abundance and Localization.
Good morning, everyone. Thank you for being here. Thank you to the organizing committee for inviting me to present some of my research on this Black History Month Research Day celebration. So I work in the roll-offs lab in the Department of Biochemistry and Molecular Biology. There we study this complex of proteins called the proteasome, which is made of 66 different proteins that come together to form a central core particle and an adjoining regulatory particle. The role of these complexes in cells is to recognize and degrade ubiquitinated proteins. So the regulatory particle recognizes and unfolds these proteins into the core particle where they're chopped up. And this material is recycled into the cell for different uses. As part of the ubiquitin proteasome system, the proteasome itself is involved in a wide range of biological events. And this is because this complex is essential for maintaining cellular homeostasis. The Protein homeostasis involves the balance between the synthesis and the degradation of different proteins at different times in the cell's life to achieve different outcomes. So for instance, during a stress response where cells may be starved, they initiate a process termed autophagy. This requires the synthesis of particular autophagy proteins and then their degradation once that process needs to come to an end. Cellular signaling is another great example where we know that particular proteins are required at different times in the cell cycle. They need to be synthesized, and then once that part of the cell cycle is complete, they need to be degraded in order to stop cell division. And cell differentiation is another example where we know some particular proteins have to be synthesized and others degraded for a cell to achieve its final fate. So we study the role of the proteasome in maintaining this homeostasis. In particular, I study how proteasomes are regulated once they've been made in cells. So a lot is known about how these complexes are assembled and the factors involved in that, but less is known about how they're regulated once they've been built. So to address some of these questions, we use a variety of assays. So here I'm showing you a couple of examples. So we can tag proteasomes with GFP to monitor their localization in different conditions. So you can see under normal conditions, most of the proteasomes are in the nuclei of these cells, but these stress conditions result in relocalization. Here they end up in the vacuole, and they're degraded, and here they end up in these cytosolic punctate granules. We can use these same GFP-tagged proteasomes to monitor how the ratio of these complexes might change with different cellular conditions, and that can inform why cells want a particular type of proteasome under a different condition, and give us a clue as to how that is regulated and why it's regulated that way. Another assay that we use is to purify proteasomes using different affinity tags. So again, we can monitor those complexes once they're purified, but importantly, we can look at the individual subunits to try and determine any kind of post-translational modifications or things like that are involved in the regulation of proteasome activity, localization, and abundance in cells. These and other assays have helped a lot in our understanding of how cells regulate proteasome abundance and localization. For instance, we were one of the first groups to show that proteasomes are degraded through autophagy. We've identified several conditions and factors that are involved in regulating that process. Proteasome storage granules, we found a lot of factors and a number of conditions that regulate and stimulate this process for the proteasome. Interestingly, mitochondrial inhibition. We're really curious to understand why mitochondria inhibited proteasome, or cells prefer to put proteasomes outside of the nucleus, and what that might mean for different aspects of biology. So there are many reasons to try to understand how cells naturally regulate proteasome abundance and localization. One example of that is different types of cancers require proteasome activity in order to survive, and proteasome inhibitors have been approved by the FDA to treat such cancers. So knowing how cells naturally regulate proteasome abundance and localization can help to inform the use of such drugs in the future. With that, I'd like to thank you all for your time. I'd like to thank the members of the Roll-Offs Lab, the department, the organizing committee, and our funding sources. I hope you enjoy the rest of the talks.
Thank you, Dr. Waite. Before moving to our next section on population health-related research, I would like to remind our audience to please feel free to submit questions to our speakers via the Zoom chat. Our next presenter is Dr. Babalola Fasharu. The title of this presentation is Collaborative Research Efforts to Improve Smoking Cessation Among African Americans.
Thank you for the opportunity to present a brief description of the work our team at the University of Kansas Medical Center has been doing in collaboration with SWOP Health Services to reduce tobacco-related disparities affecting African Americans and Blacks and improve health equity. This collaboration started over 20 years ago. Why focus on African Americans or Blacks? The graph on the left side shows cancer incidence in males by race ethnicity in the United States. Blacks have the highest incidence rates for all cancers combined and the highest overall cancer mortality rates compared to other racial ethnic groups. Blacks are more likely to be diagnosed with cancer at a late stage and are less likely to survive 5 years after being diagnosed with cancer than whites. Similarly, tobacco-related morbidity and mortality are disproportionately higher for African Americans than other racial ethnic groups in the United States. The graph on the right, comparing lung cancer deaths in relation to tobacco use among racial ethnic groups, shows that Blacks have the highest mortality from lung cancer. This is particularly intriguing in that Blacks or African Americans smoke fewer cigarettes per day compared to whites. Prevalence of tobacco use rates is about the same between non-Hispanic whites and Blacks. Over 20 years ago, the founding chair of the Department of Preventive Medicine and Public Health, now Population Health, Dr. Jasjit Aluwalia, and tobacco researchers in the department developed collaboration with the SWOP Health Services, a federally qualified health center focused on care of the underserved populations in urban Kansas City. This partnership has led to enrollment of over 3,000 participants into clinical trials to understand the biobehavioral mechanism underlying persistent smoking in African Americans. This slide shows the support from various organizations for this partnership. This include the Black Healthcare Coalition and Black Chamber of Commerce. We also had a strong support of the Community Advisory Board and the support of Congressman Emmanuel Cleaver II, who helped run some of our recruitment adverts. I will briefly describe the studies and the essential findings. Case 1 was the first smoking cessation trial that demonstrated efficacy of bupropion in moderate to heavy African American daily smokers, those who smoked 10 or more cigarettes per day. There was 17% point difference in the quit rates between the bupropion arm and the placebo arm at week 7 end of treatment, and at 26-week follow-up, the difference, though reduced to 7.3%, was still statistically significant. Other findings were that reduction in depression symptoms was higher and weight gain was less among those who were on bupropion compared to placebo. Case 2 was the first cessation trial in African American light smokers, those who smoked less than 10 cigarettes per day. 2 milligrams nicotine gum was not effective in African American light smokers. Health education worked better than motivational intervention in Black smokers. Those who smoked menthol cigarettes were not as successful in their quit rates as those who smoked non-menthol cigarettes. Case 3 was bupropion trial in African American light smokers. Cessation rates in the bupropion arm was higher than placebo, and the difference was statistically significant at the end of treatment week 7. However, there was no difference between the treatment arm and the control arm at 6 months follow-up. Case 3 also shed light on the effect of menthol on cessation. We also found that medication adherence was critical for sustained abstinence, and genetic variability in bupropion metabolism impacted cessation. Case 4 was the first controlled trial of varenicline in African American daily smokers. Stay tuned, as the results will be released soon. Quit smoking to live. The first Quit to Live study found that whites did better on varenicline than Blacks, and that socioeconomic status played a role in this difference. Quit to Live 2 did not find significant difference between counseling plus NLT versus counseling alone in non-daily Black smokers. Quit to Live 3 is ongoing. This is a multidisciplinary and multi-institutional team effort funded by multiple NIH and PCORI grants. Thank you for listening.
Thank you, Dr. Fasharu. We will now move on to our next talk by Dr. Cheryl Utley. The title of this presentation is The Efficacy of Adaptive Therapy Intervention for African American Adult Smokers, Purpose and Aim.
Each year, more than 72,000 African Americans are diagnosed with tobacco-related cancer, and more than 39,000 die from a tobacco-related cancer. African Americans experience greater difficulty quitting relative to other racial ethnic groups, despite being lighter smokers. Smoking cessation pharmacotherapy is effective for African American smokers. However, quit rates within the first 4 weeks of treatment are 14% to 36% for African American smokers, as compared to 34% to 65% for non-African American smokers. The long-term goal of this research project is to improve tobacco dependence treatment outcomes for African American smokers by identifying promising interventions. The objective of this study is to examine the efficacy of optimized versus enhanced usual care treatment for smoking cessation. The primary aim is to evaluate the short and long-term efficacy of optimized pharmacotherapy and usual care for smoking cessation in African American smokers. We have two hypotheses. The first hypothesis is that participants randomized to OPT will have significantly higher biochemically verified abstinence at week 12, which is the primary endpoint, than participants randomized to usual care. The second hypothesis is that participants randomized to OPT will have significantly higher biochemically verified abstinence at 2 weeks follow-ups, week 18 and 24, than participants randomized to UC, usual care. The design is a proposed study with a randomized adaptive treatment trial for smoking cessation in 392 African American smokers. Participants randomized to optimize pharmacotherapy, an N of 196, will receive smoking cessation counseling, nicotine patch, and up to 2 pharmacotherapy adaptations to BUP and VAR based on verified smoking status at weeks 2 and 6. Participants randomized to usual care, the N will be 196, will receive the same smoking cessation counseling and NP. Pharmacotherapy in this group will not be optimized. Pharmacotherapy and counseling in both groups will last for 18 weeks with long-term follow-up through week 26. And the primary outcome is biochemically verified smoking status at week 12. The potential impact of this proposed study moves beyond a one-size-fits-all. What works for one individual will work equally well for another individual approach. It also examines ways to time and sequence existing therapies to optimize cessation outcomes in African American smokers. This study challenges current treatment paradigms and has the potential to impact both research and clinical practice. If effective, this optimization strategy will represent a major shift in the approach to tobacco dependence treatment and have broad treatment implications for the nearly 6 million African American adult smokers in the United States. Thank you.
Thank you so much, Dr. Utley. I would like to just send a reminder to the audience that for all of you who have asked questions to our speakers and they were not addressed, they can be addressed by email. So please email the speaker if you have additional questions. We will now move to our next talk by Sierra Stites. The title of this presentation is Correctional Health is Public Health, a survey of academic correctional healthcare programs in the United States.
Mass incarceration is a public health and a health equity issue. In 2018, over 6 million people in the United States were either held in prison or jail or were on parole or probation. Now, despite these extraordinary numbers, the health of incarcerated folks is not a priority in the United States. Incarcerated people experience greater disease burden than the general population with respect to communicable diseases, non-communicable diseases, mental health disorders, and substance use disorders. The social and environmental circumstances preceding incarceration also have an impact on their health. These intersecting conditions are compounded by the oppressive and punitive environment of corrections, where individuals are often made to relinquish their autonomy, identity, and dignity. The resulting unique and complex vulnerabilities must be explicitly considered when providing health care to this population. And now, despite these concerns and despite these vulnerabilities, there's no national standards for incorporating correctional health care into medical education at the undergraduate or graduate level. With these truths and findings in mind, in 2014, Dr. Jason Glenn and his research team completed the first phase of this project. They conducted focus groups with learners at the University of Texas Medical Branch to learn about their experience as medical students and residents as they provided care to incarcerated patients. And they found that learners and attendings both require specialized vulnerable populations training to understand and not unknowingly or otherwise exploit the unique vulnerabilities of incarcerated patients. Their work inspired the next phase of the project, which is what I'll be discussing with you today. The purpose of this phase is to assess existing correctional healthcare programs at academic medical centers in the United States. We want to know if academic medical centers are talking to their learners about the health and vulnerabilities of incarcerated folks, and if so, how are they doing it? What are the current practices and what are the best practices? We designed a mixed methods research study that would be conducted here at the University of Kansas Medical Center. We identified academic medical centers with correctional healthcare programs through a literature and web search. Over email, starting back in August, I invited program directors to participate in a REDCap survey. And then with those survey participants, we followed up with an interview over Zoom or telephone. From there, we performed inductive analysis of interview transcripts using open coding, and our next step will be thematic analysis. Our response rate was 11%, which we were pretty happy with given that we were reaching out to physicians in the middle of a pandemic. Our preliminary data confirmed that there are no national best practice standards for correctional healthcare education. Our findings, preliminary findings, also reveal that if programs discuss these major themes of vulnerability with their learners, it's more likely to be done on an ad hoc basis rather than as a formal part of the curriculum. So we believe that this project will identify some of the best practices as well as the major deficiencies of current correctional healthcare programs at academic medical centers. The implementation of specialized vulnerable populations training has the potential to improve healthcare delivery, the healthcare experience of folks who've intersected with the criminal legal system, and healthcare outcomes. Many thanks to the principal investigators on this project and my capstone committee, Drs. Jason Glenn, Megha Ramaswamy, Alina Bennett, and Amber Hashmi. If you're interested in final findings, please join us on April 20th for my capstone defense. Thank you so much.
Thank you, Sierra. We will now move to our next talk by Romy Kewo. The title of this presentation is Speech Language Hearing Service Models in Cameroon.
So the problem in my research right now is there's a paucity of research regarding speech language hearing services in Cameroon. To give you some background, Cameroon is a West Central African country located on the Gulf of Guinea. The country is comprised of over 200 languages, with French and English as the national languages. There is ongoing tension between Francophones, which are French-speaking Cameroonians, and Anglophones, which are English-speaking Cameroonians. Cameroonian preschool-age children show higher instances of speech and language disorders compared to other African countries, as seen in Oyono's research. And studies conducted in Cameroon are sparse in literature focused on areas of dysphagia, voice disorders, aphasia, traumatic brain injury, or other acquired neurological disorders, as seen by Mowafu, Wankam, and Oyono. So the purpose of my research is to describe current speech-language hearing services, needs, and barriers in Cameroon. My questions: what are the service delivery models in Cameroon? How do geographic and geopolitical factors impact access to speech-language hearing services? What are emerging questions in literature specific to service delivery in Cameroon? And lastly, what are the needs and barriers of Cameroonian speech-language pathologists on how clients obtain speech-language hearing services in Cameroon? So my proposed method will entail gathering participants, which will be speech-language pathologists in Cameroon from Anglophone and Francophone provinces. Recruitment will involve contacting professional organizations, looking at special interest groups, or ASHA, or the American Speech Language Hearing Association, SIGs, going on social media, maybe personal and professional contacts as well. The measures will be presented through a questionnaire and rationale. The analysis will be qualitative analysis to determine major themes and limitations of the study. The potential application for research, my purpose in all of this research is to use this information that I gathered to improve speech-language pathology services in Cameroon and hopefully influence future directions for continued research in the area of speech-language hearing services in Cameroon. These are my references, and I want to give a huge thank you to my thesis advisor, Dr. Matthew Gillespie, for his support in this project as well. Thank you for joining me.
Thank you, Romy. We will now move to our next talk by Dr. Charlotte Smith. The title of this presentation is the Kansas Birth Equity Network.
But before I get into the details of that, I want to tell you a little history about how we started. So we started by developing a community initiative called the Kansas Sisters and Brothers for Healthy Infants. And the Kansas Sisters and Brothers for Healthy Infants are African-American graduate chapters of fraternities and sororities coming together to work on a shared objective to increase awareness around infant mortality and what it means for our community. And the inclusion of the fraternity gives us the opportunity to have the importance of fatherhood and co-parenting incorporated into that initiative. So we are a group that was developed in 2017, and each year we host an event together, and that event is Celebrate Day 366. And the opportunity to do that is to really increase awareness and understanding in our community about the African American infant mortality. You might be asking yourself, why do we do this? Why is this important? The reason it's important is because the African American infant mortality rates are very, very disturbing. So in the United States, the infant mortality rates are worse than they were in 1850. Each year, more than 50,000 women face life-threatening conditions during childbirth, and each day, 137 women suffer life-threatening complications from childbirth. But when you're Black, these are worse. Black infants are 2 to 3 times more likely to die at birth. Black women are 3 to 4 times more likely to die during childbirth. And the rates are worse than they were 15 years before the end of slavery. 15 years before there was any health care policies, before there were hospital births, and before there were workforce laws. When we look at this Kansas maternal mortality disparities, we really see that these disparities are concerning. In the state of Kansas, for every 100,000 live births, 23 white women die. For every 100,000 live births, 70 Black women die. The Kansas Birth Equity Network purpose and objectives are to develop a culture of equity between academics and Kansas System Brothers for Healthy Infants, and create a unified patient engagement program of patients and diverse stakeholders trained in PCOR, best practices, and methodology working together on research projects to promote neonatal health. The aims of this project is to recruit 50 diverse stakeholders of color, to train those stakeholders of color on comparative effectiveness research, patient-centered outcome research, and birth equity. And to do this, we have developed and piloted a birth equity curriculum. Together, upon the completion of training, we will develop a 5-year research agenda to reduce disparities in maternal and neonatal mortality. And the reason that this focus is on maternal and neonatal mortality is because we cannot address infant mortality without addressing maternal health. So one thing that we've done in the beginning of this project is we developed a red cap survey, and that survey was developed to collect maternal voices, so really specifically Black maternal voices in Sedgwick County. And the participants were asked to complete a questionnaire consisting of 5 sections: demographics, age, sex, racial identity, ethnicity, marital status, education, income, zip codes, prenatal, birth, postpartum, and health equity experiences. We also conducted a Juneteenth panel, which was a candid virtual conversation with women, fathers, clinicians, and doula about maternal and infant health. We also conducted our Celebrate Day 366 annual event, in which we focused on the state of Black families and fathers. And we developed and piloted a birth equity curriculum. So I'm going to talk a little bit about what we found in our Red Cap survey, which was our Collecting Voices study. So what we found as far as the responses for women, the number of pregnancies that were most likely were 2, the number of live births most likely amongst the group that completed the survey was 1, and we developed this initiative so that we could identify quotes. And here are some of the quotes that we found in our study. For satisfaction of care, one woman stated, "Care is not intentional when it comes to Black mothers. Our experiences, our birth stories, our physical and our mental health, because there's a lack of intention in care, we have these deployable disparities. We're not centering Black mothers, we're not listening to them, and we're not amplifying their voices." For birth experiences, one woman stated, "I noticed they treated me and my husband very badly. We had to continuously tell the hospital staff my birth plan, and they kept trying to push their own agenda." The father's experience, was stated as, but it's frustrating to watch doctors who are supposed to be there to help you and help you through this, which is supposed to be a joyous occasion because you are bringing your son or your daughter into the world, but they are dismissive. Advice from a clinician in these findings was listen to Black women, acknowledge the Black man that's in the room. Just listen to the Black woman, and that's it. That's all you got to do. If a Black woman is saying something's going on, listen to her, take it seriously. That's all, and that's it. So that is the initial part of our study, and the reason why we did that is because we want to really develop our initiative and this network around how do we address the issues in the community? And the best way to do that is to listen to the individuals that are being impacted by these issues, and that is listening to Black women. So in developing this birth equity network, one of the first starts we did was listen to Black women. Now we're recruiting those 50 members of the stakeholders of color, and we are beginning our curriculum. So if you want to learn more about us, first, our team, and I'd like to give thanks to my co-lead, Dr. Michelle Redmond, who has been very instrumental in bringing together the fraternities and sororities, and our research assistant, Kyla Morris. Visit our website for more information. If you want to join the network, please visit this link. It is a brief survey that just collects your information so that we may screen you and make sure that you have shared missions, a shared mission and goal to become a part of this work. Follow us on Facebook or contact us via email. We'd love to hear from you. The Birth Equity Network is a great initiative, and we plan to definitely make a change in the community.
Thank you so much, Dr. Smith. We will now move on to our next presenters, Todd Moore and Dr. Kevin Sykes. The title of their presentation is Taking It to the Streets, Mobile Community-Based COVID-19 Testing Program.
Hi, thank you for the opportunity to present our data here today. My name is Kevin Sykes. I'm a research assistant professor at the University of Kansas Medical Center.
And I'm Todd Moore. I'm a health policy and management PhD student.
And so what we're going to talk to you today about is our Taking It to the Streets program of mobile community-based COVID-19 testing. So this was a CDC Foundation funded grant to the Heart to Heart International, and I was the principal investigator on this study. We were funded to complete 5,000 tests across the Kansas City metro area, and we did this between May and November of 2020. So this was a mobile testing unit specifically for individuals with limited access to or opportunities for testing services. So we were trying to provide an opportunity for people who might have wanted to be tested but were unable to be tested, and specifically we increased that opportunity by adding asymptomatic individuals to the list. So we really did hone in on asymptomatics as our target population, because at the beginning of the epidemic, asymptomatic individuals were unable to be tested, and it was limited, the resources were limited to those who were close contacts of COVID-19 cases, or they were symptomatic themselves. So we did 75 events at 60 sites across the metro area and just a little bit beyond. So all the way from Lawrence up into Northwest Missouri for Excelsior Springs and Lawson. And when we focus in on the information about Wyandotte County specifically, we did 19 events at 10 different sites in Wyandotte County. And you can see on the left-hand side of this slide the number of tests that we performed at each one of these sites. But we were able to move from the far eastern parts of the county in Kansas City, Kansas, all the way out to the western parts of the county in Edwardsville.
So this slide examines the overall metro results compared with Wyandotte County residents and Wyandotte County sites. We found some interesting things here. There's a slight decrease from sites to residents, which we interpret this to be either Johnson County or external county residents who work, play, and pray in Wyandotte County. We also found more symptomatic people in Wyandotte County, regardless of site or resident. Another key finding was Wyandotte County residents are more likely to report COVID symptoms, which is significant engagement of Wyandotte County residents in vaccination. This slide shows that 73% of Wyandotte County residents who were not tested but wanted to be tested, that they're motivated and want to participate. This may be contrary to popular opinion around underrepresented minorities participating in clinical trials, and we found that Wyandotte County residents are really actively engaged in their community. This slide shows racial classifications as are reported by participants. Hispanic Latinx individuals represent nearly 70% of the positives identified by our testing program. This group is clearly disproportionately bearing a larger share of the burden of the disease, according to our testing program, and site distribution. They are also more likely to be asymptomatic positives at a rate of 12.2%, which is nearly double that of non-Hispanic populations. This slide is positive tests over time, and as you see, the positive test rate for Wyandotte County followed the same pattern as national trends. And finally, this slide is represented by resident zip code in the metro area. So here you can see a disproportionate effect on Wyandotte County, as evidenced in the darker red sections. Wyandotte County was way more impacted by this disease than any other space in the metro.
So just to wrap it all up, our program-wide positivity was 6.1%, but when we focused in on Wyandotte County residents, we reached 16.5% positivity. And it's important to note that 45% of our positive tests are asymptomatic individuals. So if you only test the symptomatic individuals, there's clearly a group of people here that are being missed. And it's unclear what that means from an infectious or infectivity standpoint, but it's certainly an important marker for the disease moving through the community. And finally, what we have also noted here is that there's an increased burden of the disease in Wyandotte County. And if that translates to an increased vaccination need, because ethically we say we want to get the vaccine to the people who are most at risk, then that should be disproportionately distributed that way, so that an additional number of vaccines are in the county as opposed to some of the other parts of the community around Kansas City. So thank you for the opportunity to present today, and thanks for listening.
Thank you, Todd and Dr. Sykes. Well, our next presenter, and actually this is the last presenter of our short talk, is Dr. Consuelo Wilkins. The title of this presentation is COVID-19 and the Faith Community, an exploratory look at pastor, faith leader, health communication among African Americans.
Hello, and thank you for tuning into our presentation titled COVID-19 and the Faith Community, an exploratory look at pastor, faith leader, health communication among African Americans. My name is Consuelo Wilkins, Associate Professor in the Department of Family Medicine and Community Health Research Division, and my collaborator is Dr. Yvonne Chen at the School of Journalism and Mass Communication. The purpose and background of our study, unfortunately, we know that African American men and women carry one of the largest burdens for COVID-19 deaths in the United States. And those numbers are even greater in Kansas, where African Americans are 6 times more likely to die from the virus, and rates in Missouri are not too far behind. So there are multiple determinants that are reported for these disparate numbers, including testing access, utilization, housing, discrimination, and several other factors. The rationale for this study is based on research that we have conducted, where African American community and faith leaders are trusted individuals within the community, especially here in the Midwest, and they may be appropriate health communicators and promoters of culturally relevant public health information. And so we wanted to explore pastor and faith leader beliefs, opinions, and their knowledge about COVID-19, and whether they saw themselves as viable channels of this type of information. And we also wanted to give them an opportunity to acquire some additional skills and health communication training in the spirit of community-based participatory research. The method that we used was a qualitative method where we followed a CBPR approach that informed our study design and our recruitment. So we conducted 11 in-depth interviews that were faith leaders and pastors from the bi-state area, 5 from Missouri, 6 from Kansas. Most of the sample was male. And the analysis is preliminary analysis and also data results. There were two of us, myself and my collaborator, that did the preliminary analysis. And in that preliminary analysis, the results revealed that there was an overall disaffection toward the state and federal government's lack of unified communication efforts. So as a result of that, these pastors and faith leaders actively sought out information from their pastoral community networks and other resources such as the CDC and the local media to educate their congregation. They also highlighted recommendations for how governmental agencies could improve their communication efforts. And some of those recommendations were being transparent, communicating the knowns and unknowns. And then the recommendations for the content was adding messaging of hope to existing communication, adding testimonials and relevant Bible scriptures and so forth to enhance message receptivity among faith members. So implications and conclusions include that COVID-19 messages can be improved by transparency, inclusion of spiritual language, also messages of empowerment and hope. And faith leaders and also pastors saw themselves as leaders who protect their congregation's mind, body, and soul, especially during the pandemic, and made recommendations on how those messages could be improved through delivery and also content. And in the spirit of CBPR, we also held a health communication workshop for the first cohort of the 11 last year and are targeting to have another one this spring after we have an additional number of individuals to be interviewed in the second cohort. So we'd like to say a special thanks to the consortium. Without the help of members, would not have been able to recruit, and then also to the Department of Family Medicine and Community Health and other individuals who are listed here on this slide. Thank you.
Thank you, Dr. Lumpkins. Well, that concludes our short talks. So now let's please thank all of our speakers for taking the time to share their research experience with us today. I would also like to give a shout out to those who have been working behind the scenes to ensure that everything runs smoothly. Specifically, Antoinette Redmond for taking care of all the planning logistics for today, as well as our student volunteers and our two sign language interpreters. There is a polling for today's research activities that you can find the link to the REDCap survey via the chat between the keynote presentation and the closing remarks. The organizers would really appreciate you completing it. Now we will take a 5-minute break. We will see you back shortly.
Hey, Dr. Wilkins, how you doing? Oh.
Hi, Dr. Wilkins. How are you?
I'm doing great. How are you?
Great. Thank you so much for joining us.
It's my pleasure to be here.
Hey, Dr. Wilkins, how you doing? It's Todd.
Todd, hey, how are you?
Good. It's been a long time.
It has been so wonderful to see you.
So happy that you're able to join us and finally got you to be a part of something here. Been trying for years to get you here.
I know. It's I'm right now. I'm looking forward to visiting everywhere. So
yeah, amen to that. Amen to that. Well, we're glad you're here.
Yes. How's the morning been going so far?
Good, very efficient. Antoinette's done a wonderful job coordinating everything. Man, is she such a blessing.
It's great.
Well, also the committee has been wonderful, and Diane has been leading us as well. So it's a bunch of people all together.
Sure. That's true. That is true.
So does the entire group hear us speaking? I just saw somebody comment. I thought I went to a breakout room. So
I think we're on a break right now. So
okay.
And then we'll come back and
we're all on a break, but we can hear you speak and we can see you.
Yeah.
Mm-hmm.
Oh, great. Okay. Well, I guess I won't say so much about how I wish I was there to have some barbecue, but
no one will be mad about that. I think it's we're all like wanting to get back out. We're getting closer.
Okay, good. Well, I do know that Antoinette has been keeping everybody on track, including me virtually. I'm getting used to that. Broderick keeps me on track also.
He keeps all of us on track.
Yes. So I was following instructions to get on early enough, but I'm going to take a quick bio break if I may before we start. Is that okay?
Oh yeah, absolutely.
Absolutely.
Okay. All right. I'll be right back then.
How are we doing with time, Antoinette? Are we good?
We're ready.
We're ready?
Yep. So I'm just going to pull it back up and we'll get ready to rock and roll.
All right. I was planning on looking at the clock, the countdown, and I was like, I don't see it. So
switch it up really quick.
That's okay. All right. So we're back. All right. Well, welcome back, everyone. And our remaining segment will be moderated by Todd Moore. So let's welcome Todd.
Hello, everyone. Thank you, Dr. Mahoney. I'm proud, honored, and humbled to serve as your moderator for the second half of this research day. As Dr. Mahoney said, my name is Todd Moore, and I'm currently a PhD student in the Health Policy and Management program here at KU Med Center. I've also served as a research project director in the Family Medicine, Family and Community Health Research Division for over 8 years and a founding member of the Black History Month Planning Committee. And as a tribute to Dr. Thomas, the Research Day Planning Committee commissioned a wonderful video commemorating Dr. Thomas's impact and influence here at KU Med Center and the region. So we want to show you this video, then I'll make a few short remarks.
My name is Crystal Lumpkins, and I am an associate professor here at the University of Kansas Medical Center.
My name is Grace School. Right now, I'm working in the School of Pharmacy at Rutgers University.
My name is Nikki Chang. I'm an associate professor in the Department of Pathology and Laboratory Medicine.
I am Lisa Van Hoosen. I'm at the University of Louisiana Monroe, where I serve as the founding director of the Doctor of Physical Therapy program.
My name is Michael Moncure. I'm a general surgeon, trauma surgeon,
and I am now at Truman Medical Center. I'm a professor of surgery there and like vice chair of the department there. Prior to going over to Truman about 4 years ago, I was here at KU.
So I met Dr. Thomas back in 2008 when I was applying for a position here in the Department of Pathology.
I met Dr. Thomas in 2010, and that was actually a very fortuitous meeting because I was volunteering at this campus.
It's probably in the early 2000s where I had the opportunity to hear her speak, and I was captivated.
I had never even been to Kansas City before, never. And between a couple of friends and individuals within the city and within the institution, no, I was kind of lured here to take a look. And when I looked at what was going on, I was pretty fascinated by the fact that they had a faculty development program for underrepresented minorities or underrepresented medicine individuals. And it was being run by Ira Porter and Dr. Patricia Thomas.
There was no Black History program. There wasn't all of the different affinity groups. All of that, and I'm not saying that she was the only one, but she was a mighty force and one of the people that facilitated that.
My first impression of Dr. Thomas when I first met her was that she was a vivacious, passionate person who loved what she did, and it showed up in all of her being.
She was just a force to be reckoned with.
I mean, she was just committed to so many people. And it just spoke to me. It reminded me of, the quote that talks about, how one person can change the world. And that was definitely Dr. Thomas.
Her legacy and her work, I mean, say a lot. I tell students now, when I talk to KU students, when I talk to Truman students about what you can create by just being an example, being a partner, being someone that's looked up to by the university, someone that is touting the message that the more diversity, the more equity, the more inclusion there is, the better off the institution's going to be. You can make a huge impact, and she was able to do that.
It was such a proud moment that I was able to give the first Patricia A. Thomas Diversity Inclusion Award to Pamela Scott, who is now one of the directors in the Office of Diversity, Equity, Inclusion for the School of Medicine.
She was just very calm, always very thoughtful, always have a good plan, and always be there to help the mentees. So I was like, wow, how could a person, I guess being a pathologist helped, but I was very impressed by that.
I knew I wanted her to be my mentor. I knew I wanted to sit at her feet and learn. How does one live a servant life in that way?
Mentorship was a core part of who she was. And I have been able to take what she taught me, and I've been able to share that with dozens of people, dozens of medical students.
She has such a big impact on a lot of junior female faculties at KUMC.
I am so happy that the university, this program has chose to remember her legacy.
Thank you, Dr. Thomas, for the fact that I was an individual who had a lot of passion and a lot of desire to be successful in academic medicine, but I didn't have a model.
I so much appreciate what you gave me. It was simply priceless.
By you being an example for me, it's allowed me to be an example for other individuals.
Thank you for taking the time to mentor me. You've been a wonderful role model.
You gave me the ability to stand when I didn't think I had any strength. You gave me the ability to go on.
Wow. What a tremendous video for all of us, and the effect she had on all of us. Servant, leader, model, mentor, guide. It's so wonderful. In 2014, Dr. Thomas approached me about assisting her with an off-site event she called the Black History and Health Summit. I helped her with that event, and you see the cover page here. That was a successful event. We had over 150 people attend, and so we plan to conduct an on-campus event the next year. She passed away suddenly in 2015, as many of you know, and I knew we had to continue to build on that on-site event she would be proud of. Dr. Jennifer Keaton was the Director of Special Projects and Strategic Initiatives in the EVC's office at the time. I talked to her about my idea of doing this on-campus event, and she thought I was a little crazy to take this on, but fortunately for me, she liked crazy. So thanks to the support from Dr. Keaton, the Office of International Programs, SNMA, and the Diversity and Inclusion Cabinet, we went on to convene Black History Month after nearly a 20-year hiatus, and that original framework has served as a model for ethnic research recognitions campus-wide. Dr. Thomas felt it was vital to tell the history of Black History Month at the Research Day event and to serve as a stanchion of where we started and a beacon of where we want to go. Next slide. So many of you know the story of Carter G. Woodson. He was the son of former slaves born in Virginia in 1875. He escaped poverty through education, and after earning a doctorate degree from Harvard, he dedicated himself to filling important and significant gaps left in American history books through developing and advancing the field of African American history. He pressed for the respect and recognition of Blacks in the shaping of America, and in 1926, he initiated Negro History Week to formally recognize and celebrate these contributions, choosing February because it is the month that marks the births of both Frederick Douglass and President Abraham Lincoln. It is in Woodson's honor that Black History Month has been celebrated in this country since 1976. And it is in Dr. Thomas's honor we celebrate it here at KU Med Center. Dr. Thomas would say Black History Month is not about just one group of people or a culture in isolation. It's about the social, political, and economic impact of a people, the African-American experience, and our unique discoveries and tremendous influences in the shaping of this country. As you saw in our presentations earlier, we can have an impact in science and on campus, in our community. Do you agree? Raise your fist in the air. Turn your cameras on. Raise your fist in the air. There we go. Black History Month is also about unifying the community, not just the Black community, but the American community. It is therefore more than appropriate that we come together to think about and talk about Black health, Black healthcare, equity, and justice, not only at KU, but in Wyandotte County, the greater Kansas City community, and beyond. If you agree, raise your fist up. Put them up. We're gonna call and response all day, y'all. Okay, here we go. Next slide. This year marks the 57th anniversary of the Civil Rights Act of 1964. 75 years since Bayard Rustin and George Hauser's original journey of reconciliation in 1947. That first journey heralded subsequent freedom rides, from Rosa Parks in 55 to the Black Lives Matter marches and murals here in Kansas City last year. We are in an ongoing and unparalleled fight against unequal practices, discriminatory policies, and systemic racism. We've come a long way, but we have a long way to go. A long way to go. Do you agree? Raise that fist up. All right, next slide. Dr. Thomas believed that an academic medical center can and should cast a positive halo effect over its surrounding community and engage in partnerships that create outcomes that are greater than the sum of their parts. Dr. Thomas believed that we together can take up a new journey, a journey toward collaboration, discovery, promise, and reconciliation. She inspired me to continue her effort to identify, recognize, and elevate the spirit of Carter G. Woodson here on our esteemed campus. Black History Month began as a way to forge partnerships where they were not, uncover the hidden, shed light in areas of darkness, and show strength in areas previously weak. In the spirit of Dr. Thomas, Carter G. Woodson, and the countless others who came before us, we continue the effort of our ancestors to build awareness, overcome barriers, and recognize our contributions to the American culture. Do you agree? Come on, bring them up. So I declare to you, I am a cisgender African-American man from Junction City, Kansas. My wife is from Kansas City and a graduate of Lincoln College Prep in Kansas City, Missouri. My youngest son was born here in Kansas City, and I embrace KU as not solely my place of employment, but a part of that community, the community of my birth, my wife's community, and the only home my youngest son has ever known. Dr. Thomas shared these sentiments to me and with me, and KU Med Center has always been a home for her for many years, a place where she experienced loss, challenge, discomfort, and grief. She also thought of it as a place of promise, potential, opportunity, and hope. As a family, I do as well. Do you agree? Raise your fist up, hold them up. My charge to you today is to commit yourself to find someone to help achieve their goals and aspirations. Embolden them with your courage and determination for social justice and health equity, and become the light you want to see in the world. In truth. Thank you. Rest in power, Dr. Thomas. All right, thanks everyone. Now it's time for the Dr. Patricia A. Thomas Keynote Address. Dr. Geraldine McGee, the Vice Chancellor of Diversity, Equity, and Inclusion here at KU Med Center, will introduce our speaker. Dr. McGee.
Absolutely. Thank you, Todd, for that tribute to Dr. Thomas, but also your words of encouragement and our charge as we go forward. It is my pleasure to introduce our keynote speaker. Dr. Consuelo Wilkins, a trained geriatrician, is Vice President for Health Equity at Vanderbilt University Medical Center. She is also a Professor of Medicine and Associate Dean for Health Equity at Vanderbilt University School of Medicine. Dr. Wilkins is a nationally recognized thought leader in health equity and in addressing elimination of systemic inequities that impact the health and well-being of racial and ethnic minorities. As a community engagement research scientist, Dr. Wilkins has pioneered new approaches to engaging vulnerable, socioeconomically disadvantaged, and minority populations. Dr. Wilkins is the principal investigator of 3 NIH-funded centers focused on translational science, precision medicine, and disparities. Dr. Wilkins is also the principal investigator of a Robert Wood Johnson Foundation Award on engendering trust in healthcare among African American men. Please welcome our keynote speaker, Dr. Consuelo Wilkins.
Thank you so much, Dr. McGee, for that kind introduction. And I'm really absolutely thrilled to have the opportunity to spend some time with you all today. Hard to come after Todd telling everybody to get their fists in the air. So I guess I better dig deeper before lunchtime today. It was really inspiring also to hear the to see and hear the video about Dr. Thomas. I am honored to be able to deliver this keynote today, and certainly inspired by what she poured into so many. So thanks again for the invitation. Thanks to the committee, to the planning committee, to the research day committee, to Dr. McGee, Dr. Zhu, Dr. Mahoney, and others who I'm sure many have been involved in planning today. So I'll start with that I was given really specific instructions on what I'm supposed to talk about today. So I do appreciate that. That's typically helpful. And there's so many things that we could talk about as it relates to Black history. And I, again, do appreciate having some direction about what you'd like me to cover. So I was told I should talk about mentoring students, faculty, and staff. And again, I take these things to be in some ways literal, but also open to my interpretation. So, you know, mentoring, I changed to engaging. And I intentionally will try and talk about students, faculty, and staff, although I will admit that my knowledge about mentoring staff is, I have less of that personal experience. So just want to give that as a, at the outset. I know we're offering some continuing education, so just giving my disclosures here. As Dr. McGee pointed out, I do have some research funding, and that's listed here, NIH, PCORI, Robert Wood Johnson Foundation, and also more recently a project that is partially funded by the American College of Radiology. I will also just want to talk about what I hope to cover today. And by the way, you all will help me cover this. It is challenging still to give these kinds of presentations virtually without, you know, being able to interact with the audience. So I'm going to ask you all to interact with me and use the chat box. And if you have questions, if I say something that doesn't sound right or is confusing, or you want to chime in, please use the chat box. I'll do my best to try and keep up with that. Instead of mentoring, I'm saying engaging intentionally. Some of you I know know me from the world of community engagement. And so that is an intentional word, because I do think that mentoring tends to be unidirectional. It's, you know, one way approach. And if nothing else today, I want us to leave making sure that we recognize that for all mentoring relationships, I mentor quite a few people, and I always gain something from that. And I think most people who mentor do that. But I think the real value of engaging people of color needs to focus more on just what that personal value is, but for those of us who are in leaders, especially in academic health centers, need to use that personal change and those different ways of thinking to dismantle the structures that have caused us to have these inequities and the lack of diversity to start with. So we have to move from this community service deficit mindset that we're doing something just to help people of color, and really recognize that this is how we're going to solve the huge problems that we currently face. The problems that we face around health inequities, the lack of real innovation. We spend so much money on healthcare in the United States, yet we have these gaping holes and huge disparities in how people fare from our health system. If we're really going to erase these deficits, eliminate these disparities, save money from the healthcare standpoint, we have to have a workforce that looks more like the people who live in this country and in the world. So I'm going to try and go a little bit over some of the historical factors that are really the reasons that we have this underrepresentation, and talk about some of the biases, but also I want to spend some time talking about what we should be doing to successfully engage people moving forward in a way that is mutually beneficial, so that people get what they need to thrive, but also we start to dismantle these systems that have kept people oppressed for too long. So it is Black History Month, and so I want to make sure we start with celebrating Black excellence. I wasn't here for the entire program, so I'm not sure how much of this you did already, but I think there's so much for us to celebrate. And so sometimes we hear about the same you know, a couple of dozen history makers. So I wanted to point out a couple that you may not have heard about. George Carruthers, Dr. Carruthers, who you see here pictured with his, you know, pretty sci-fi looking device here that's from actually the 1960s, early 1970s. Dr. Carruthers grew up in a single parent home. And if you look at when he got his degrees, he was born in 1939. He just recently died last year. At the time when he is getting his Bachelor's of Science in Physics and Nuclear Engineering, and then later his Master's and PhD in Astronautical and Astronomical Engineering. This is before, you know, the Civil Rights Act was passed. So imagine, you know, a man of color doing this kind of work at a time where in some places in the country, he couldn't vote or wouldn't be allowed to vote. And so these barriers that are in front of him are not just, you know, hurdles, as I have a slide later. These are roadblocks, people coming, things coming out of nowhere to keep him from being successful. Yet here he is next to his invention, which was a part of the Apollo 16 mission, really used to visualize things in space and on the Earth in a way that no one had imagined or had only imagined at some point. Here's Dr. Lillian Holland Harvey, who is well-known, a well-known nurse educator, really responsible for educating many, many Black nurses who, of course, are still underrepresented in terms of the biomedical and clinical workforce today. She was Dean of the Tuskegee School of Nursing back in 1948. So just wanted to share a couple of examples that you may not be so familiar with, and then want to take a step back. So I'm talking about Black excellence. It's Black History Month. Why are we celebrating Black excellence? For those of you who are here, I'm sure most of you this is preaching to the choir. You know why we're celebrating Black excellence. You know about the issues, the oppression, the marginalization that communities of color have experienced, especially people who are descendants of enslaved people in the United States. But not everybody understands that. Not everyone recognizes the importance of doing that. And part of that is, you know, again, this deficit mindset that often people of color have been marginalized and oppressed and thought of as people who just aren't high achieving. So somehow this celebration of excellence is because of, you know, myth of meritocracy, and that somehow, you know, those who have been able to be successful just, you know, are worthy of celebrating because there's something pathological or damaged about being Black or brown. And that is a narrative that unfortunately exists that we aren't addressing well enough, that we continue, and that's going back to the why I think it's important for us to not use this deficit mindset of just mentoring because it's the right thing to do, because people need some community service of some kind, when we have to recognize that the structure that we live in, the systems that we function in, are intentionally keeping people out. And that's also intentional in this language of historically excluded. We often talk about underrepresented. Yes, underrepresented is an important term because we're comparing to population, but we shouldn't say underrepresented without recognizing that people have been purposefully, intentionally excluded in the past. We could spend the entire day talking about, how the American Medical Association did not allow Black physicians to be included. We could spend the entire day talking about the medical schools that did not allow Black or brown students to be admitted. We could spend many hours talking about the reports that closed most of the historically Black medical schools. We could spend a lot of time talking about these things, but more importantly, we need to recognize that those did not happen in isolation, and we are still experiencing the long-term impacts of those decisions that were made, those exclusionary practices. And let's be honest, some of them still exist. They have not completely gone away. So these are some of the challenges that we know people who are from communities of color have experienced and how they've been historically excluded, things that they have to grapple with. We were not included, we were not allowed to be educated before, so there are fewer of us in positions of power, fewer of us teaching, fewer of us in academic health centers, period. The ongoing disinvestment in our communities, lack of wealth, lack of opportunities, all of those things compounded are disadvantages for us, but we still continue to experience marginalization. So, for those of you who are, and I'm not sure how many of you are not in within the medical center, but for those of you who are in an academic medical center, for me, I can count the number of Black women and Black men in my department who are full professors on my fingers. So that isolation, marginalization, those experiences with micro, macro, overt aggression are front and center. These are things that we cannot escape. And that's after you've actually had the opportunity to be educated in it and have the opportunity to work there. So let's talk a little bit about history. So I'm not going to attempt to talk about Kansas City and specifically because I'm sure I will get something wrong. But redlining has happened across the entire country. So a time in our history where you could not buy a home, you could not get a mortgage because certain communities were designated as undesirable, primarily because that's where Black people lived. So lending opportunities to, again, own your own home. This is how wealth was built. And so these are things, again, that happened all over the country in the 20s to the 60s. And so some people will look at things like redlining and they'll say, well, that was decades ago. That's a common thing that we hear about, why are you still talking about, Black history and the need for inclusion and all of those things, because people keep saying, well, that was a long time ago. Well, here's the example from Nashville with redlining, and this was published in a local paper last year that shows the redlining that happened in the 30s and 40s still has an impact nearly 80 years later in Nashville. So those same communities that were considered to be undesirable, and there was lack of investment. So think about that. If you didn't have a home, you didn't have, if you couldn't own a home, where the schools are the best, where the grocery stores are put, where the businesses are, all of those things, where the dumps are put, where there's this exposure to toxic environments, are more likely to be in these neighborhoods that are already labeled undesirable. So we have to keep all this in context as we're talking about the people who need to be included in our health center. So also, like many places in the country, the interstate had an unfortunate took an unfortunate toll on the communities in Nashville that were already disproportionately impacted, communities of color. So the building of the interstate, which took many years, actually, interestingly, ended up a case that went to the Supreme Court because people in the Black community in Nashville recognized that this was going to disrupt their community, which, by the way, despite all of these challenges, was thriving. So the resilience of our communities, to be able to have Black-owned businesses and to actually create communities and some wealth, despite all of those barriers, then took this huge hit after the construction of the interstate basically disrupted the community. You know, we have several historically Black schools in Nashville, including Meharry Medical College, and disrupted the community such that, you know, these are effects that we still see in the neighborhood, despite now some gentrification that is now pushing out communities even more. Okay, so this is going to be, I see some comments. If there are any of these that are questions, Dr. McGee or Antoinette, if you would, you know, wave at me or let me know, or interrupt, please let me know. Here's my first question to you all. Okay, so we're talking about redlining, we're talking about you know, where people live. These are actual heat maps of where people who work at Vanderbilt live. And so there are two different maps with two groups of people. So the red is, if it's red, that's where more of higher concentration of people live. And if it's green, there are fewer there. Okay, so anyone want to guess, you know, what you're seeing? So people who work at Vanderbilt, these could be, you could tell me that these are African Americans, Hispanics, you could tell me they're nurses, you could tell me they're doctors. And so on the right, since you don't know Nashville, where this little central area is, that's the core of Nashville, and it's very close to Vanderbilt University Medical Center. So the little red dot, like really close to the medical center, is on the right. And then on the left, you have people who are spread out all over the region. Any guesses? If you want to use the chat box, that'd be great. No, it's not fair because everybody doesn't know Nashville. Highest mortality rates. Okay, Jason, where are the highest mortality rates? Which one, left or right? Left and red. Okay, that's a great guess. Although this is not what this is intended to convey, you are correct that there are higher mortality rates, lower life expectancy in the map on the left, which is where we have more of our service employees, which is also where more Black and brown people live. So here in North Nashville, this is historically Black part of town. You don't get a prize, sorry. There's no prize, other than being right. Isn't that prize enough? So here, these are areas in town where more Black and brown people live. These are areas in town where we tend to have a higher proportion of Hispanic, Latino, New American people who speak languages, primary language is something other than English. But I want to make this point here. Here's where our faculty live. So more of our physicians, more people who have a higher, you know, earning live here close to the medical center. So if you are here living close to the medical center and you are making decisions about policy, if you are mentoring people who live here and you don't understand their the barriers that they face just getting in to from the medical center every day, if you don't understand how long it takes them to commute, if you don't recognize that these people cannot use the medical center based childcare, one, because it's more expensive, two, because they can't get to it in a timely manner, especially if they have to use public transportation, then you are not really understanding the barriers that people are facing. And so that's part of, again, what I'm saying with the, you have to understand what the policies, practices are that need to be changed. And those are things that you need to learn if you're mentoring people who are from backgrounds that have been historically excluded. Now this is shifting a little bit to what we're talking about with race. So we're talking about communities of color, and which one is, you see the numbers next to the color. These are literal colors from a like a painting a scheme or a color scheme here. Which one of these is white? And which one of these is black? Dr. Glenn is not coming through for us on this one. Does anyone else have a response? Which one of these is, yes, not, okay, there's some people coming through. No, none of them, exactly right. So, so why is it that we refer to, we're talking about Black History Month, we're talking about Black people, we're using the term race, and we're none of those people are Black or white. So why have we, defaulted into this extreme of color? So it's as if we polarized race intentionally. Yes, that is correct. So, I got some good ones coming through here. Isaac is talking about the 1 drop rule. Yes, so somehow 1 drop of Black blood, and by the way, I'm still waiting for someone to tell me what a drop of Black blood looks like. And we're in academic medical centers. Can somebody identify that drop of Black blood and tell me what it looks like? Because I don't think we know how to figure that out. But these social identities, and I think it's really important, at least it's important to me. I identify as a Black woman, and that's my cultural identity. However, it is not biological or genetically based. You cannot tell me that I'm not a Black woman, because that's who I am. But you also cannot tell me that because I identify as a Black woman that I am somehow inferior. Yet that is the narrative that has been set up for us over time with this social identity. So if you look at how the race ethnicity designation has been used since the 1700s, 1600s, it's been for the purposes of oppression. So there's the 1 drop of Black blood means that you're Black. Again, that makes no sense at all. So why was that done? To keep people from having biracial, or for there to be intermingling, for people who are enslaved to continue to be marginalized. Similarly, the opposite was true if you were American Indian or indigenous person, that you had less rights if you were sort of told to stay to yourself, so that you could, if you wanted to keep any of your any of the citizenship rights that were going to be afforded to you in your own country. So lots of things with this, how we determined, who's identified how, and quantums and all of those things. So you have less access to funds that were given or set aside for your people after your land was stolen from you. Still to this day, in 2021, the United States government uses a question that only acknowledges 2 ethnicities. You're either Hispanic or you're not. And certainly one of my pet peeves that we use way too much in medicine and health is to describe someone as non-Hispanic. It's as if, you know, the last thing you want to do is be identified as Hispanic. So let's put that clarifier in front of you. But you know, we're not acknowledging the many different ethnicities that exist. You know, Asia is a continent, Africa is a continent. There are many, many different ethnicities of people in these continents, which, by the way, account for about 70% of the world's population. So most of the world is either Asian or of Asian or African descent, yet somehow we put people in these huge big buckets and presume that we know something about them based on these categories. So I'm going to talk a little bit about racial equity, and I'm going to try and frame this in terms of engaging and that bi-directional mentoring and exchange of information. So, you know, our goal, at least my goal, is to make sure that every person has a fair and just opportunity to be healthy, and more broadly, you know, a fair and just opportunity to reach their full potential is what we think about with racial equity. And as I've been trying to highlight some of these, you know, structural reasons that people aren't able to achieve their full potential, we have to get back to the systems in place that were put in place to marginalize people. Now, I want to ask this question because it's really important when we talk about mentoring, when we talk about opportunity, when we talk about test scores, when we talk about you know, how we write job descriptions that get people into positions, all of those things, is our educational system. So why is it that in a city, your taxes for, you pay taxes you pay go for everything, and infrastructure goes to the city government, goes to the roads, goes to the water system, goes all these different things in the city. But somehow when it comes to education, your tax dollars go to a very specific zone for schools. So you have schools in the most affluent communities that are heavily resourced, that have lots of money, and then schools, probably those that are defined as hazardous when during the time of redlining, have lower budgets, fewer resources. Why aren't those resources, why aren't all those property tax dollars going to one central budget, and then every school gets what it needs for the students that need to be educated? Why is that? How could that be? Capitalism is not benefiting. Racism. Okay, Jason, I'm gonna turn yours off. You gotta let other people weigh in. All right. To oppress people, exactly. So if we really wanted the best for all of our children, we would be advocating for those dollars to go to be used and distributed centrally. Now, this is a button that I think that pushes people who think of themselves in some ways as social justice minded a little bit, because then you start talking about their kids and what they have access to. Like, well, that you're going a little too far now. You want me to share my resources from I pay a lot of property taxes. I should keep that to myself. Okay, well, sounds like you really don't want everybody to thrive. You're riding on the same roads as everyone else. So why aren't we all sharing in the pot of dollars to educate all of our children? These are policies that were put in place to disadvantage and marginalize and keep some people oppressed. And if we don't begin to dismantle and remove these policies and practices, then we're going to keep thinking about people of color in ways that make us seem to be pathological in some way, that we have this burden of such a dire need, when what we really need is to not be excluded. We need for things to not be kept from us. We need to make sure that information isn't siloed. We need to make sure that when you talk about our communities, it's from the lens of understanding these structures of oppression are still present. So, okay, I'm still depending on you, Dr. McGee, to tell me if I need to stop and address something here. But I want to spend my last little bit of time of slides before I open up to questions, talking about what's next. So this is a slide I showed before. And so I want to talk about some of the strategies, and I want to hear what you all are doing as well to engage, again, not just mentor, but to engage. So we know that there are so few faculty of color in medical centers, but there's certainly a dearth of health professionals, period. And so, how are we overcoming the issues of lack of exposure? I think exposure is really important early on for people to be able to see themselves as someone. And, some of the things that we're doing here in Nashville just recently are making sure that people don't just see physicians and nurses, because that's if you're watching television or you've not had exposure to different health professions, that's typically all you see. What about the radiology technicians, and what about the physical therapists and occupational therapists, respiratory therapists, the pharmacists? There's so many health professionals. There's so many people who are touching other people's lives. Your research day is focused on discovery. All of the things that need to be discovered with a lens that's not going to further marginalize communities of color. We need to make sure that people have access to and can see that. I think until we get to the broad exposure, the increased numbers that we need, we have to make sure that everyone, and so I shouldn't say until, but both. My mentors early in my career were mostly white people. My primary mentor was a white man. My secondary mentor was a white woman who were brilliant in their fields and had the humility to understand and know that a lot of what I wanted to do, they didn't understand. They didn't understand health disparities. They didn't know how to get into the community, but they were willing to help me, work with me to get access to people who knew how to do those things. And so I think that level of humility in mentoring is really critical to ensuring that the people who need you get what they need and not just what you have to give. Because if we are only getting what you have to give, then we're going to be stuck in this, cookie cutter, keys made from the same cloth, everybody in the same lab, acts the same way, and is prepared to do the same thing, but we're not actually preparing people to do something differently. We need people who think differently and can approach issues and solve problems differently. Now we could spend, again, the entire day talking about issues related to prejudice and discrimination and those silos that are created, but specifically around mentoring, we have to recognize that everybody is having a different experience. And if we're not open to understanding those experiences so that we can change things, then we're going to be stuck. So that's, again, this deficit model where if you're mentoring, if you're like, especially in the last year, since in the last 10 months plus, you know, this heightened awareness of racial injustice, the many other murders, unfortunately, that we've seen in the hands of police in the last year, have a lot of people stopping to think, along with the COVID inequities, you know, we've got to do better, we've got to do better. And so there's this pull for people to do more, but still this pull is, again, more of that, I have, so let me help, as opposed to, I have some things, but I'm missing some things. And there are things that I need to know and I need to learn. And that's where we get to this, you know, the need for this bidirectionality. The value of our differences are not deficits. And, you know, one of my biggest pet peeves when I'm talking to colleagues is that when they're talking about hiring or bringing someone into a lab or mentoring, like, I don't think there's a good fit. Like, fit is the excuse for us not wanting to change something. So if you expect people to fit into a mold, then you're not ready. If that mold is all you can do, then you got some soul searching to do. You need to be prepared. And I think we in leadership at academic medical centers have to help people get prepared and help people get comfortable, help people get uncomfortable. I tell my colleagues, especially around community engagement, that, you know, if you're not uncomfortable at times when you're doing community engagement, you're not doing it right. Because if you're doing it right, then the community finds you trustworthy enough to tell you what you're doing wrong, to give you some feedback and some input, to let you in on something that can help you change things. You're not doing it right if it's always comfortable. And so we have to focus on, you know, valuing these differences, recognizing that difference is not a deficit, and that what we learned in this mentoring process as we help people get to a place where they feel authentic in their jobs, that they have a sense of belonging, then that's when we get to the intentionality of how do we identify, you know, these exclusionary practices? What kept you away? One of the questions I like to ask people when I'm talking to them is, you're important, you're special, you're remarkable, but who do you know that you think was just as remarkable as you, or close to it? You know, some of us are a little arrogant there, but didn't get this opportunity. And I think about that a lot because I grew up in a small town in the Mississippi Delta, and I had some incredibly smart friends and classmates who did not go to college. And my thoughts about them are often like, yes, I'm special. Yes, I'm great, but I'm not that much more special or great, and maybe not as even special or great as some of them, but they didn't have that opportunity. There were more obstacles, there were more barriers, there were more challenges for them. And maybe there was a little bit of luck for me. So how do we change that narrative to say, okay, well, my cousin Anthony didn't get this opportunity. This was a problem for him. This was a barrier for him. The test taking skills, the biased tests, the number of kids who have test prep that makes it seem like they're, you know, doing so much work. Like, there are lots of these things that we have to reframe that don't focus on this deficit mindset, but focus on the assets. I went to Howard, historically Black medical school and undergrad. When I got to Duke to do my internship and residency, I had some of those fears about not being as prepared as my colleagues who were from Duke and Harvard. But when in the middle of the night, when it was time to put the NG tube down a person's nose, I could do that better than any of them because I had some of my rotations at DC General Hospital. I could talk to patients and get information. They trusted me, they shared information with me. So those should be considered as assets. And so when we're asking these questions and we're checking off boxes, we have to be able to look for these different skill sets, characteristics, things that people bring to the table. And we have to give them as much credit as we give everything else. We often leave the story part out of our approach. We value numbers. We value only data that we can count. We don't value the stories that people bring to the table. And I think that is something that we really have to change if we're going to improve the way that we deliver care, the way that we conduct science, the way that we actually understand how we can change our health, our lives, and that of our community. I think I'll stop here and just say that, the final point for me of the value of mentoring, the value of engaging communities of color is not just for them. It's not politically correct. It's not because we've not done well in the past. The value is that we're going to have a better, higher functioning, thriving society. We are all going to actually be more successful if we do this right. So thank you.
Okay, wonderful. Thank you so much, Dr. Wilkins. Let's give her a raised fist. Turn your cameras on, raise that fist up. Thank you for your time today. We have some time for some questions, I believe. And Antoinette and committee, please correct me if I'm wrong. Do we have a couple questions we wanna shout out to Dr. Wilkins real quick, one or two? Go ahead with her. I'm hearing someone on mute. No.
Well, I thank people for using the chat. I said use the chat, but then you all just started chatting. I couldn't keep up. So
we're good about that here, actually.
Yeah. I love it. I love it. I love it. I think, that call and response, I certainly missed. So I appreciate that.
We do actually have about 10 minutes for questions. So I encourage everyone to either put it in the chat or take your mic off of mute, because we have Dr. Wilkins here and she has given us a great presentation, and we certainly want to get everything out of her that we can. And so I would encourage you to ask questions or comment. I thought it was a great presentation, Dr. Wilkins. Thank you so much for what you shared today.
I see a question in the chat from Letty. Okay. So maybe Letty, can you unmute and maybe ask your question in person?
Sure, can you hear me okay?
Yes.
Thank you so much for your presentation, Dr. Wilkinson. I was commenting about one thing that I've noticed working in medical centers is that we see a lot of this excellence from our Black, Indigenous, and people of color groups who are able to make it out of that wonderful image that you showed. And so my question is, how can we contribute and make sure that we engage with communities where these people come from that not everybody made it out of because of these systemic issues that you just presented today?
Yeah, thank you for the question. I think it's really important to recognize the individuality in the people who are able to make it across the alligator, crocodile, barbed wire, fencing. Some people are ready to share, want to share. Some people want to be in these roles where they're helping increase the diversity. And some people just want to do science. They just want to take care of patients. They don't want to focus on that. And I think we have to respect that, and we have to find better ways of allowing people that space to do so. Because again, not everybody wants to do it. I will say that I love what I do, But this wasn't the career that I want. I was going to study Alzheimer's disease and focus on, African Americans, preventing them from getting Alzheimer's disease, identifying it early, and all of those things. But the work that I needed to do in the community, there was no one doing it, and there was no one engaging people and getting those people into clinical trials was really challenging. And so, I think we have two big buckets of things that we have to do. We have to figure out how to make sure that everyone has that opportunity to do the work they want to do. Everyone has the opportunity to be in medicine, healthcare, research, public health, whatever they want to do, and that we move those barriers for them. And then we have to find ways to actually dismantle the system that is continuing to perpetuate, race as something pathological, as a deficit, and not as a culture to be celebrated. So I think there are many different approaches, and some have been, really successful. But what will work is probably going to need to be multi-pronged. I think that's been part of the challenge. We just want to try one thing at a time with one community, and people are very different and they have different needs. I do think that, again, from my own personal background, a lot of my friends and family, their parents didn't go to college and didn't know how to navigate the systems, and were worried about, how to navigate that system. But of course, they couldn't do it without money. The pull, the power, the lack of dollars. I think there's so many challenges. So if there are ways for us to individualize that approach, I think that would be good.
Thank you, Dr. Wilkins. I have a couple of people queued up for you.
Okay.
And then we'll get those two questions in. Dr. Utley, I see your fingers go up, so I'm going to assume you want to ask a question. So I'm going to put you as well. But Juliana, do you want to ask your question?
Yes. So can you hear me well?
Yes.
Yes.
So I found very interesting the model of conscious inclusion. We hear so much about conscious bias, but not so much about inclusion. And I wonder if there is any type of training for mentors who want to mentor minorities, because we know it's good for their CV to mentor minorities and have inclusion in their teams, but I wonder if there is any type of training in order for them to do a good job.
We desperately need it. There are a couple of mentoring programs. There's one Angela Byers at Wisconsin leads, culturally aware mentoring, that I think is really good. It's intensive, and I think that's part of the challenge, that people want to be able to just flip a switch and gain these skills, but a lot of it requires you to do some internal work, and that again gets people uncomfortable. It takes time. And it's a long-term process. So there are some good programs. I think we need more. I would love to see more of them funded and available and present at institutions across the country. I'm trying to keep it short, Todd, so I can get to other questions.
Okay, great. Lisa Van Hoos. Dr. Van Hoos, you had a question? Go right ahead.
Yes, and thank you so much for an amazing presentation. My question was do you have any recommendations on terms other than community of color or BIPOC? Because there's a lot of discussion right now about how those are still centered with white being the reference point. So I didn't know if you knew of any new terms that might be on the horizon.
Yeah, thank you. I think it is it's something that's obviously evolving, as you said. I try to define the terms when I'm using them. I would say even in my own house, I don't know that we agree on Black versus African American. So it can be quite challenging. That's why I included historically excluded, because I think it's a broader term because it doesn't give a label, but then it also lacks specificity. So I think there are some challenges there. But if we're talking specifically about the workforce or the medical center, then really acknowledging that these are people who are not just underrepresented, but they have been excluded. And some of that might not have been intentional by the people who are in positions of power now, but at some point it was intentional that Black people, brown people were not allowed to be educated, were not allowed into college, were not allowed to read as slaves. Like, those are intentional things that we have to acknowledge as this is excluded. People were excluded.
Thank you so much, Dr. Utley. Go ahead and ask your question. I think this will be, we have time for this will be our last question. Is that correct, Antoine? Okay, great.
Okay. So I like to fast forward, and I like to hear your perspective during this COVID-19 pandemic, and what should our role be in helping to alleviate the mistrust within African American communities regarding vaccinations, or what should be done in terms of health equity?
Thank you. I'm spending quite a bit of my time right now doing this work with the vaccines. I think one thing I learned from the community, should not say vaccine hesitancy. I should talk about vaccine readiness. Do communities have the information they need to make a conscious decision? And have we given time to answering questions? So my approach has been, we're working with churches, we're working with community organizations to actually say, okay, this is going to be the time for you to ask questions of a health professional. And it's my job to be trustworthy and answer those truthfully. I think that's really what we need to do, is make space for people to get ready and make that decision, but also come back when people say, I don't know, I don't have enough information. I need to see it play out a little bit more. We can't just leave it and move forward.
Thank you.
Okay, everyone, let's thank our keynote speaker today, Dr. Wilkins, for taking time to join us. Our Black History Month Research Day celebration with a raised fist, put them up. Thank you, Dr. Wilkins. Now we have Dr. Abiodun Akinwutan, Dean of School of Health Professions, to deliver the closing remark. Like Dean Ojo, Dean Akinwutan is an excellent researcher with an active study program here at the Med Center. Dr. Akinwutan.
Well, can you all hear me? Okay, good afternoon. And thank you very much to our last presenter, Dr. Consuelo Wilkins, and thank you for the fantastic presentation. The day started with introduction to activities of the day, and then we heard the insightful and nice opening remark by Dr. Akinlolu Ojo, the Executive Dean of the School of Medicine. And then after that, we had several very impressive research presentations by our distinguished scholars and researchers. We should be truly proud of the works and accomplishments of our colleagues and be appreciative of the life-changing research activities they engage in. Please join me by a raise of hand in acknowledging their accomplishments and their presentations. Thank you to all the presenters. And then we listened to a befitting tribute to Dr. Patricia Thomas for all that she represented. Finally, we all listened to the insightful presentation by Dr. Consuelo Wilkins. This has just been phenomenal. This has been a great research day and one of the hallmark celebrations of this Black History Month. Before I say a final thank you, I want to introduce you or talk about Shady Tana Muscle Alexander. She was the first African-American to receive a PhD in economics in the United States. and the first woman to receive a law degree from the University of Pennsylvania Law School. She was the first African-American woman to practice law in Pennsylvania, following her father's footsteps. She was the first national president of Delta Sigma Theta sorority, serving from 1919 to 1923. And she is the first of so many things. And she has this inspirational quote, a word that I quote, and I think this applies to all of us. Do not let anything stop you. There will be times when you will be disappointed, but you just cannot stop. I thank you all for your participation today. A special thank you again to our keynote speaker and all the presenters. To all those who have been working behind the scenes to make sure the Black History Month is the best activity we would have to celebrate this, and for all their contributions behind the scenes, we say a big thank you to everyone. And I wish us all the best as we continue to celebrate our history, which I hope will not be restricted to just 1 month, but a daily activity and something we continue to do every day. And I will end my closing remarks with the words of the legendary James Brown. I am Black and proud. Thank you.
Well, say it loud, brother. I appreciate you, those words. Dr. Akinwutan, it should be noted that Dr. Akinwutan was the first dean to participate in Black History Month Planning Committee and consistently makes time to be a part of the committee planning process. And I appreciate that. I know Dr. McGee appreciates it in the research, the planning committee leadership appreciates that as well. So thank you so much.
Thank you.
So before we adjourn, just know that we're gonna, there's still some more events coming up. They're listed there. That's a couple SNMA events are coming up, and then, or several SNMA events are coming up. So look for those, including one featuring our very own Dr. Jason Glenn. You may want to check that out, and all the rest. Thanks so much to the committee, the Research Day Planning Committee. Thanks to our co-chairs, Dr. Lynette Sparkman-Barnes and Margaret Ogaruna. Oh, Margaret, dang it, look, I know, I'm sorry.
Okanaran.
I have to look at it to say it. I couldn't see it. I don't have your last name memorized like that. I apologize, but know that there's love for you, and I appreciate your leadership for Black History Month. You and Dr. Sparkman-Barnes, and of course our ever-present leader, Dr. Jeremy McGee, for making this and elevating this event to where it is today. I really appreciate all of y'all. Thank you, and we're adjourned.